This topic we're going to be discussing protein folding, and in and of itself this could be many weeks of study, but we're just going to briefly go over the topic and have you understand a little bit of how proteins fold and how we measure those, so you can understand that proteins, based on their primary structure, have the information to fold, but they can misfold and sometimes need assistance in vivo to assure proper folding, and that they're stable enough but not too stable that it doesn't prevent them from being rapidly degraded. So we're going to go over those particular points in this topic, and the learning objectives are described below. So the key thing to remember is that the primary sequence determines its folding arrangement. In other words, it's the sequence itself that will determine the protein's folding arrangement. And for some proteins, you don't need assistance by other molecules, other proteins, but some proteins you do. So this fact of the primary sequence being important, the primary structure, was done by Christian Amundsen around the 1960s, and he was awarded the Nobel Prize in 1972 for his work. So we can think of an energy landscape where you have free energy here, and this is your native conformation, and that when proteins undergo a folding process, they can adopt various levels of stability in their folding as the ensemble gets funneled down to the native conformation. There may be some intermediates that are formed that need some assistance, but primarily the amino acid sequence of the residues will convey the folding arrangement at some point, but there could be some intermediates that are formed that will have to be assisted in a way to make sure they get to the native conformation. Okay, so Amundsen, he used ribonuclease A, which is a small protein that has 3 disulfide bonds, and he basically what we call denatured the protein by providing urea to unfold the protein and reduce the disulfide bonds with a reducing agent to make them free sulfhydryl groups, and then dialyze those components away and allowed the protein to refold, and did many studies to look at the protein refolding process. So one way to assay whether the protein was folded or not was to see if it could do its catalytic activity. And if it could, then you knew it was folded properly. So that's just a quick rundown of the overall experimental approach that was used to look at trying to understand these protein folding steps and intermediates in the process. We're just going to briefly go over the concepts here of protein folding and their stability. So to study protein folding, we also, study their denaturation. We look at how proteins denature. And so a way to do that is to have a protein that's folded, and we can denature it in several different ways. And one way would be to use a combination of thermo and chemical means to do so. What do these experiments provide? Well, we're trying to understand protein stability as it relates to the free energy. So that would be the free energy of the folded protein in the system that exists in, which is some kind of aqueous system. And we have components of enthalpy and entropy. And so we have to consider the enthalpy of the bond breaking and formation from an unfolded to a folded protein, as well as the entropic components of the system as a protein goes from unfolded to a more ordered structure. And so the interplay between enthalpy and entropy is going to give rise to this delta G, whether it's a favorable or unfavorable protein folding process. And so How do we do that? We do these protein denaturation experiments. So a protein is completely folded, and then we're going to crank up the heat. And so we're going to heat up the protein. So if we're looking at proteins in our system, they like 37 degrees Celsius. But what happens if we get higher temperatures? What happens to the proteins? Well, as we increase the temperature, the folded state becomes less stable. You're starting to increase the vibrational frequencies between hydrogen bonding interactions, and these hydrogen bonds are going to break, and your secondary structural elements are going to start to fall apart. Your ordered water molecules are going to start to dissipate away, and so the system is going to become less stable to support the folded protein. It's going to unfold, and if you crank up the temperature high enough, the protein is now an unfolded polypeptide chain. And so the transition that occurs between 0% denatured to 100% denatured at 50% mark, the halfway point, the transition is what we call the melting temperature. Now, this melting temperature does not mean that half the protein of the molecule itself, that half the protein molecule is unfolded and half of it's folded. It's an all or nothing process. That means at the 50% denaturation, half the molecules in the population are unfolded and half remain folded. Because the energies are of stability are so small, it doesn't take much for the protein to unfold. So you're not going to have partially unfolded proteins. They're either going to be folded or not. If there is a deviation in this curve from the ideal behavior, it could be a 3-state model instead of a 2-state model, folded and unfolded. It may be folded, intermediate, unfolded. So it's possible to have an intermediate state that has some kind of a folded structure, but it's not a native folded structure. And it's not an unfolded structure either. But we're going to consider only the case where it's folded or not, a 2-state model. Another way to promote the interaction or the unfolding of a protein is to add a chaotropic agent. And this is an agent that can be used to break hydrogen bonds between the secondary structural elements of folded proteins. And so they act as themselves to break up the interactions that help stabilize proteins. So urea would be one of those, guanidinium chloride. You can add more hydrophobic things like detergents that have or amphipathic. They have a polar head group and a nonpolar tail. The nonpolar tail can promote the unfolding of proteins by providing a hydrophobic environment that will allow the protein interior to become exposed to solvent. Alcohols can do the same thing as well. The other way to do it is to change the pH. We talked about before how a protein charge state will be dependent on the pH because of the acid and base groups that are present on the protein. And so extreme pH values can also be used to cause proteins to unfold as well. Primarily we'll use some kind of a chemical or thermal denaturation process. We can also go the other direction. We can expose our protein to one of these chemical denaturants like urea, and you need high concentrations to promote unfolding. So like 8 molar urea or 6 molar guanidinium chloride would be required to promote protein unfolding. And if you want to resolubilize the proteins, you basically dialyze away the excess reagent. You basically put your sample into a dialysis tube that has a membrane permeable component that allows small molecules to go across the membrane, but the protein stays in the tube. And you can dialyze and put that into a beaker that has no urea, and the urea will leave the tube into the into the beaker, but the protein will still stay in the tube, and you're changing the environment to be less denaturant and more conducive to folding. And then you can study after a certain period of time, take out your dialysis tube, look at the protein in there, and see if it's still active. This is kind of what Amundsen did with his experiments with ribonuclease A. So these are called refolding experiments because you refold the protein after unfolding it. So here's some experimental ways to look at protein denaturation. So we're going to take the protein and we're going to expose it to a high concentration of urea, as shown here. And as you see in this experiment, they're electrophoresing the protein under different urea concentrations, and the higher it goes, the migration changes, and you see this transition at the 50% position would be at this inflection point, supporting a two-state model. We can also do the experiment using spectroscopic means to look at protein denaturation. So recall we have tryptophan that could be monitored. And if tryptophan's on the surface or buried, it will have different spectroscopic properties, and we can follow that with fluorescence or UV vis measurements. There's also a technique called circular dichroism that can help us look at secondary structural elements of proteins. And so if those secondary structural elements are no longer there, we're going to change in the circular dichroism measurement, and we can monitor that for secondary structural element changes as well. So if you were to do and superimpose all of these data, you can see they all fit upon this two-state model. So this is how we look at measuring protein denaturation, and the inflection point here would be our point where half the population is folded, half is unfolded. So we can actually derive an equilibrium constant from the denaturation curves as shown. So when we look at the equilibrium, we're looking at equilibrium between the native and the unfolded version. And so as we denature, we're going to get more of the unfolded versus the native structure. And based on what's shown in the plot, in terms of defining the fraction folded as 1 minus alpha and the fraction unfolded as alpha, we have this relationship, and the delta G of unfolding, or I should say the delta G of folding, is minus RT natural log of KEQ. So you could derive the KEQ in this fashion by measuring the different amounts of the unfolded and unfolded state, and then we can get a delta G if we want to of that from the KEQ. So we have the values here of 1 minus alpha and alpha at any given time. We can determine that, a urea concentration, and then we can get a delta G value accordingly from that. To plot that urea concentration around that transition to 50% folded to 50% unfolded in terms of the population, we can extrapolate all the way back to 0 molar urea, and this would be our protein stability here. In this case, we're looking at 5.6 kcals per mole. We're used to kilojoules, and we'll get back to kilojoules in another table, but it's an older one. So this is kind of how you can figure out protein stability from these curves. We're not going to do that in this course. I just wanted you to know that that's how the data can be used to identify protein folding stability by extrapolating back to 0 concentrations of your chaotropic reagent to find out what the stability of the protein is at that position based on measuring the delta G from the KEQ at other urea concentrations or chaotropic concentrations. Okay? So what you end up with something that looks like this down in the lower right hand side is a protein, the conditions that you did the renaturation or denaturation, and a delta G of folding for the reaction. And so you can see here these are kilojoules per mole, and you can see they're negative, so that they're going to favor folding, and that these values vary, but they're not a lot. 7 kilojoules per mole, 32 kilojoules per mole, 57 kilojoules per mole. And so think about some of the thermodynamic values we had for, say, ATP hydrolysis in the cell, maybe 58 kilojoules per mole. So you're looking at stability of a bond for one of the more stable proteins versus the strength of a couple hydrogen bonds for like ribonuclease, or one strong hydrogen bond. So say you have a hydrogen bond that's around 10 kilojoules per mole. Well, the stability of ribonuclease is one hydrogen bond. So gosh, that's not very stable, is it? Well, there's a reason for that. And if we look at the thermodynamic considerations involved in protein folding, remember our delta G is delta H minus T delta S. So we have to figure out those contributions of enthalpy and entropy for the whole system. That includes the protein and the surrounding aqueous environment. So I like this graph here, it illustrates it very nicely. When we look at enthalpically, in terms of bond breaking and bond formation, it takes energy to break bonds, but when you form bonds, you gain energy. So in this case, the enthalpy is negative because we have a lot of internal interactions in a folded protein that give rise to a lot of bond formation. Van der Waals interactions, charge-charge interactions, polar, dipole-dipole, all of those non-covalent associations we talked about in a folded protein. Hydrogen bonding, of course, with the secondary structural elements. There is a lot of bonds being formed in a folded protein. When you have the hydrophobic groups come together in the core of the protein, you are breaking some of the water molecule hydrogen bonding because you're freeing up that ordered water molecules that were used to accommodate those exposed residues. They're now being buried in the protein. Those are gone, and so you do lose some of those bonds. Those bonds are broken, but you have a lot more formed. So when you look at the difference between the hydrogen bonds and the water that have to break because the hydrophobic groups are forming in the core of the protein, the other favorable contributions to enthalpy in the protein are much greater than what's lost through water hydrogen bonding being broken. So you have an overall negative delta H there. And then when it comes to the disorder of the system, the entropy, you do have a very ordered protein, right? It's no longer unfolded, but a folded three-dimensional structure. So that's a lot of order. But because of the hydrophobic effect, you see, we have a lot of free water molecules because of that hydrophobic core formation in the folded protein. So that helps to offset this contribution of more ordered system in the folded protein with the more disordered water molecules in the solvent, combined with the enthalpic contributions of forming all those hydrogen bonds to form those secondary structural elements, that the overall delta G is negative enough. It's negative enough. It's negative enough. Notice lactoglobulin and 5 molar urea is not going to be folded very stably. Get the urea out of there, it'll be more favorably folded because urea is helping to unfold it. So you can see that protein stability is, around 25 kilojoules per mole on average. You're not going to have a lot. So you're just talking about a couple hydrogen bonds here and there in terms of stability of the overall protein. And so that's why when we do these protein denaturation experiments, we consider the halfway point as a two-state model, and that 50% of the population is unfolded and 50% is folded, because it doesn't take too much energy for it to completely unfold. It's a very cooperative process. And so this allows proteins to be, to be unfolded rather easily. There's not a lot of energy required, and that's important because we need to degrade proteins when they're not performing as they should and reuse the amino acids for other purposes. And so if they were too stable, there would be a huge energetic component required for its unfolding, and that would require a lot of additional energy, like I said, to do that, which would not be a good utilization of ATP because we need it for other things. So the thermodynamics favors folding, but just enough to keep it stable and to do its job, but easily to completely unfold it and use the protein for other purposes. When we look at in vivo protein folding, and that's kind of shown here as the protein chain is growing as it comes off the ribosome, as it reaches the mRNA, that domains will start to form as it's emanating from the ribosome and eventually form a stable protein. But often there could be a requirement where some of the components that are down and haven't been even made yet have to interact with a previously existing domain in such a way that if it's folded in this manner, there's going to be some misfolded regions. It has to kind of be denatured a little bit and refolded. And so something has to help denature this and help it to refold. So we're not going to have chemical entities in the cell to do this, but we're going to have proteins to do this, called chaperones. And these chaperones are multimeric units of proteins that help other proteins to fold. So this first one that was characterized was called GROEL, and this was in E. coli. This is an example of a chaperone. There's a lot of different ones there. It was found by doing some protein separations on a polyacrylamide gel, and they looked at staining these proteins with a stain, so you can see a dot there, and they saw that when they increased the temperature, they saw new proteins appear. And they said, well, what are these proteins? And so they isolated them and characterized them, and they were called heat shock proteins because you heat shock the bacteria, you see these proteins come up. And this protein, when people study it, found that it actually binds to hydrophobic regions of proteins. And there's a hydrophobic cavity that allows that protein to refold. And so this is an example of a chaperone. There's different types of chaperones, and this is the one from bacteria. We have chaperones as well. And so I just want to go briefly over how this process takes place. So you can think of these as our proteins that recognize hydrophobic residues of misfolded proteins, and it helps them to refold. So what it does bind the hydrophobic regions, and it uses ATP hydrolysis. That's the energy required to help the protein to refold through conformational changes in the proteins. We'll talk more about these conformational changes in ATP hydrolysis and other examples in this course. But you can imagine the ATP hydrolysis, which does provide a lot of free energy, can be utilized to move the protein around a little bit and to help this assist this protein to chaperone it into its finally properly folded native conformation. So briefly, this GROEL recognizes the unfolded protein, and with the help of ATP and GROES, which is another component here, which kind of caps the top of this once it captures the protein in there, allows these conformational changes to occur through ATP hydrolysis that promotes the protein to be folded. So basically it captures the unfolded protein, binds ATP and GROES, kind of caps the one end. It allows the protein to fold through ATP hydrolysis, and then a displacement then of losing ADP allows the molecule to be displaced in the native protein to be exported and back into solution. And so the chamber of the GROEL provides this hydrophobic environment, allowing the protein to refold under those conditions. Again, it's all part of its amino acid sequence that's there to allow that a chance for that protein to refold under those conditions that are different than the aqueous conditions on the outside. So that's the concept there. It provides an environment to help the protein properly fold to its final native conformation. We've got structures of the GROEL, GROES complex, and so people studied a lot of it for many, many years, decades. I even read about it in graduate school and did journal clubs on the topic as well. And so I just wanted to provide some structural aspects of it. So if you cut away and look on the inside, you can see these nice chambers, these cavities here that have these areas for proteins to get in and to refold. I like this slide right here. This shows the electron density map from cryo-electron microscopy. So this is where you use electron microscopy at low temperatures to get protein structure by taking thousands and thousands of photographs of or micrographs of the structures in solution, and then computationally put them back together to get a three-dimensional structure. And so shown here, you can see the T4 coat protein GP23 from bacteriophage, which is basically a bacterial virus. And you can see that it's denatured here in the trans ring, and then in the cis ring, you can see it's been properly folded into its native conformation. And then it will be exported out of that. So you can kind of see evidence of the actual recognition of the unfolded version on the one side of the molecule, and then the folded version on the other end. So the GROEL exists as a dimer, and so binds the one native denatured at one end. The folded one leaves this one, cap this one, fold this, then it would leave, and then the other denaturation would come in. So this captured the point in which the native protein was completely folded and ready to be released, then capturing the denatured one, showing evidence of structural evidence here supporting this folding process mediated by GROEL cavities of the GROEL complex. So I think that's pretty neat. So misfolding of proteins is actually involved in a variety of diseases. For example, cystic fibrosis, there's a mutation in a calcium channel that it's misfolded, so it can't function right. You have cystic fibrosis from that. When we look at Alzheimer's disease and amyloid fibrils that can form, or amyloid plaques, this is again a misfolding of the beta amyloid because of hydrophobic groups that are exposed and cause aggregation and plaque formation. So this is kind of interesting in terms of these fibrils that are present, in terms of how the proteins mature and can be induced to form fibrils. And so some of my current research now, I'm trying to develop methods to look at fibril formation and using novel chemical labeling methods and mass spectrometry analysis to try to understand some of these early events to fibril formation and get some insights and do some modeling to try to understand how this aggregation, this misfolding can occur. And if we can understand that, we can maybe use something to prevent this aggregation and to stop it from happening. So there are a variety of proteins that undergo misfolding and aggregation events that are related to disease. And here's just a table describing some of those: Alzheimer's disease, Parkinson's disease, spongiform encephalopathy. You have that. You also have it in terms of traumatic brain injury due to concussions. So there's a much more knowledge about concussions in athletes and giving them the proper rest and time needed for their brains to heal from such a concussive impact to their brain before they are subject to potentially another concussive impact. Because what happens over time with these concussive impacts and brain damage is you get these plaques forming. And so the brain starts to form a lot of these different plaques, and this is called chronic traumatic encephalopathy. And so again, the change in the protein structure to promote aggregation through the exposure of hydrophobic groups and residues can lead to a variety of deleterious effects in organisms. And so it's a very interesting area of research. It's hard to figure out, but I have some ideas and maybe hopefully get some funding to maybe do this for latter part of my research career. So we're kind of looking into some different ways into looking at some of these misfolding diseases. So that's just a brief introduction to protein folding or unfolding, if you want to look at it that way. But to get an idea that proteins are stable but not too stable, they can be easily denatured, and we can take advantage of the fact that we can degrade them in our cells so we can utilize the amino acids for other purposes, and that if proteins are misfolded, chaperones help to identify them and to help them fold properly. But sometimes misfoldings occur through mutation or some other change in the cellular environment due to some kind of an injury, and that aggregation can occur, and that can lead to deleterious effects in the cell. So you're not going to be doing a lot of calculations on delta G's or KEQs or anything like that in this section, but just wanted you to take away some key things of understanding protein folding in vitro versus in vivo, and some of the thermodynamic considerations of what makes a protein form a stable folded structure.